Good afternoon. I'm so sorry we're a little bit late. It's my pleasure to welcome you to this afternoon's webinar on behalf of the Biochemical Society and Portland Press. We're really pleased to welcome you all today to this webinar, which is part of our Biochemistry Focus webinar series. The topics in this series include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give you an opportunity to ask questions via text, and we welcome suggestions for future topics and speakers for future webinar series. So my name is Christine Foyer. I am Professor of Plant Sciences at the University of Birmingham, and I am hosting today's webinar with my colleague, Professor Ilse Kranner, who will speak to you in a few minutes. Today's webinar is titled Plants and Climate Change, the Role of Plants in Achieving Net Zero, and it is a taster for the forthcoming plant collection in the Biochemical Journal. So Ilse and I are co-editing this special issue on plants, and we welcome you all to submit articles for inclusion in this plant collection. You can publish for free with the Biochemical Journal. The journal is supported by an expert editorial board which adheres to the painless publishing philosophy. So painless publishing means that we get articles through to publication as quickly as possible without endless rounds of revision, one hopes. All of the profits from the journal are returned to the Biochemical Society to support its charitable endeavours. The theme collection will focus on the biochemical plasticity and adaptations of plants and plant microbial interactions to climate change. These are important because they underpin crop and forest resilience to climate stress. Plant productivity and ecosystem processes are all supported by these processes so that nutrient cycling and carbon storage can take place. The collection will not only feature advances in knowledge of underpinning plant and microbiome biochemistry and molecular mechanisms, but it will also highlight our ability to understand and predict these impacts and how climate-driven shifts in plant productivity and plant-soil interactions will occur. We will consider manuscripts, reporting results from laboratories and also from field experiments. We will take a range of topics from biochemistry in model and crop species, but also the root-associated microbial communities. So we will welcome a wide collection of papers. The submission deadline is the 30th of September 2022. And so I will hand over to my colleague, Professor Ilse Kranner, who will now introduce our speakers.
Thank you, Christine. Welcome, everybody. I'm Ilse Kranner from the University of Innsbruck, I'm Professor of Biology. And at today's webinar, we are delighted to welcome three invited speakers. Dr. Yude, USDA Forest Service, USA, followed by Professor Alexander Ruban, Queen Mary, University of London, UK, and Professor Wolfram Beckwert, University of Vienna, Austria. Before I introduce the first speaker, I would like to inform you that questions will be answered after all the speakers' presentations, but you can send in your questions during the talks. If you have a question, please type it in the question box as shown in the image on the screen, and we will try to answer as many as time allows at the end of the webinar. Right, so it's my great pleasure to invite our first speaker, who is Dr. Yude, USDA Forest Service, USA. Dr. Pan is currently a senior research scientist with the Climate, Fire, and Carbon Cycle Sciences Group of the Northern Research Station, USDA Forest Service, and senior investigator of Harvard Forest, Harvard University. She's a member of the US government's Carbon Cycle Science Steering Group, member of the Global Forest Expert Panel on Biodiversity, Forest Management of the United Nations, member of editorial boards of several important journals such as Ecosphere and Ecological Applications. Yude's expertise is in biological responses of terrestrial ecosystems to multiple environmental stress factors, with emphasis on understanding complex interactions among biotic and abiotic processes and between ecosystem structural and functional dynamics. Yude's research is highly interdisciplinary in nature and has integrated intensive field studies with modeling approaches, combining strength of both process and statistically based methods with hierarchic observations and measurements from forest inventories, eddy flux towers, and remote sensing to understanding how ecosystems function across multiple scales. And now, without much further ado, I would like to hand over to Dr. Jude Pan, please.
Yeah, I'm ready, right? Hello, everyone. Recently, the world has converged on the goal of achieving net zero greenhouse gas emission by 2050. To achieve this, there is a need for negative emissions of about 3 to 6 gigaton carbon per year to compensate for inability to eliminate all emissions from fossil fuel. So it is important to know how much carbon sequestration our natural ecosystem could contribute for curbing atmospheric CO2 and solving climate crisis. So this is the most recent global carbon budget. Hold on, let me use. So from here, we can see that carbon emission from fossil fuel, emission from land use change and land use and land cover change, and the carbon absorbed by ocean remained in the atmosphere. So the imbalanced carbon is supposed uptaking by the terrestrial ecosystem. That's a terrestrial carbon sink, which is about 3.4 gigaton carbon. So this group reported the global carbon budget every year, so which shows that terrestrial carbon sink has been continuously enlarged over the last 2 decades. In the meantime, quite a few remote sensing-based studies suggested that globe has been greener or been greening. So this map shows that the leaf area increased on most lands over the last 3 decades. So those studies suggest that the driver for greening is likely the elevated atmospheric CO2 concentration. We think this global greening and the enlarged terrestrial carbon sink could be somehow related. So in our recent studies based on the data from 14 CO2 enrichment experiments, we found that CO2 enrichment in the woody and the grassland ecosystem have very different response patterns to annual precipitation. It shows that CO2 enhancement of above ground productivity increased with annual precipitation in woody ecosystem, while decreased in the grassland ecosystems. Although different responses within ecosystems annually, if on the ecosystem average and across multiple ecosystems spatially, both woody and grassland ecosystem have a great enhancement with a drier baseline climate. We also found there's a close relationship between the enhanced leaf area index and enhanced above ground MPP. So this relationship fits for the different vegetation type. So what these results means, They suggest why all the ecosystems show the relatively high CO2 enhancement in the drier locations. Locally, woody ecosystems such as a forest have a great CO2 enhancement in the wet years because of their flexibility in the leaf area expansion with abundant water supplies. Because this equation was empirical and derived from CO2 experiments, it means by using it, if we know changes in LAR, we can estimate changes in vegetation productivity affected by high CO2. With available global LAR datasets, we can make a global application of the equation. So this map shows LAR changes between the 2000 and the 2015. Over the time, 69% of vegetated land have enhanced LAR and 30% with decreased LAR. The areas with decreased LAR are mainly caused by disturbances such as intensified wildfires in the boreal zones, which left the fire scars. The effect of worsened drought in some very dry areas, such as semi-desert of Kazakhstan, Brazil, Campinas, and this cold desert. Human disturbance, such as deforestation around the Amazon rainforest, Congo River, all resulted in the loss of LAR. However, many dry land have a high increase in LAR, such as in the Mediterranean regions, India inland, and Australia open woodland. Especially, a high LAR enhancement appeared in the temperate zones across continent. The zones include not only temperate forests, but also major farmlands and ranchland, such as Great Plains across over US and Canada. In response to changes in the LAR, above ground NPP have enhanced in most area over this 2000 to 2015. Especially 86% of vegetation had enhanced AMPP and 14% with decreased AMPP. Even some area with a slight decrease in LAR, there could be still some increase in AMPP because of direct effect of high CO2 on photosynthesis at leaf level. Again, especially temperate zones have more AMPP enhancement. Here's a summary table about the relative changes in LAR and AMPP based on ecosystem types. Globally, LAR increased about 10%, AMPP about 18% over the study period. That is, LAR increased by 0.6%, while AMPP 1.1% annually. So this summary table about above ground NPP and the changes, these are real values. So global NPP was changed from the 54 gigaton carbon in the 2000 to the 63 gigaton carbon in 2015, gaining 8.8 gigaton carbon annually. So among the ecosystems, evergreen broadleaf forest, mostly tropical forest, had the highest above ground production, followed by the savannas systems and the grassland, because these two types of the ecosystem have the vast land. Although evergreen broadleaf forest had the smallest enhancement ratio in AMPP, they still gained gained the large enhanced production, about 1 gigaton, because of their high productivity. The small enhancement is because these forests are more saturated with LAR and have less room for leaf expansions. Forests altogether gained about 2 gigaton carbon production enhancement, while the savannas, the grassland, and even the cropland also gained a larger amount of AMPP enhancement. So here there's an issue about land classification, because this study is based on the MODIS land cover class, which defined the forest as with the tree cover greater than 45%. However, the FAO definition of forest is the land with the tree cover greater than 10%. So if we use FAO definition, most of woody savanna and the part of savanna would be classified as the open forest. So these dry open forests appear to have a great potential for enhancing terrestrial production. So now let's back to the earlier question about terrestrial carbon sink. Because above ground NPP is only part of total NPP, that's about 60%, 67% on average. After big carbon loss of the heterotropical respiration, the left is a net ecosystem production, which is about 30 to 40% of NPP. And the final term for the carbon sink actually is net biome production, which is about 40% of NEP. So after these conversions, enhanced NPP equals about 1.8 gigaton carbon enhancement in the terrestrial carbon sink. So this number matched very well with the estimation by the global carbon budget. So here are a few conclusions. So over the 2000 and the 2015, global LAR was enhanced by 0.6 and AMPP by 1.1 annually, and 68% of vegetated land had increased LAR, while 86% had increased AMPP. The major driver for LAR and AMPP enhancement is likely the elevated atmospheric CO2 concentration. Sorry. Temperate zones achieve the more greening and AMPP enhancement because of the room for LAR expansion and adequate growth condition. Evergreen broadleaf forest had smaller LAR and AMPP enhancement ratios than other ecosystems, but manifest large gains in AMPP because of their high productivity. Boreal forests were enhanced in LAR and AMPP by high CO2, but scarred by intensified wildfires on the changing climate. Due to increasingly saturated LAR, intact and dense forests that played roles as capital terrestrial carbon sink in the 2000s appeared to give way in the 2010s to open forest, woodland, shrubland, and savannas, which were benefited by improved water conditions on the high CO2. However, of this ecosystem, time for preserving carbon in storage could be much shorter. Okay, that's it. Thank you.
Thank you very much, Yude, for this impressive insight into your work. And now I'm pleased to introduce our second speaker. Our second invited speaker today is Professor Alexander Ruban at Queen Mary, University of London. Alexander Ruban obtained his PhD from the Institute of Photobiology in Minsk, Belarus, in 1987 and worked until 1990 in the Institute of Plant Physiology in Kiev, in the Ukraine. In 1990, he received the British Council scholarship to work at the University of Sheffield for a year, and at the end of this term was appointed as a research associate at the Department of Molecular Biology and Biotechnology. In the year 2000, he was awarded the qualification of a university professor in France. And from 2002 to 2004, he was an invited professor in Paris. Since 2006, he has been working in the School of Biological and Chemical Sciences at Queen Mary University of London as a lecturer of biochemical chemistry, of biological chemistry, and from 2009 was awarded a title of Reader in Biophysics, and in 2010, Professor in Biophysics. His area of research is photosynthetic light harvesting. He was one of the initiators of several European research networks that focused on studies of the processes and molecular mechanisms of light harvesting and photoprotection in plants. The capture of light energy by photosynthetic organisms is a global process with efficiency reaching 80 to 100%. Alexander's talk will introduce you to the organization of a natural light harvesting machinery, the antenna of the oxygen-producing photosystem II of higher plants. He will explain the key principles of its design and provide example of how plants can efficiently and safely capture light for life of our biosphere. Alexander, the floor is yours, please.
Thank you very much, Ilse. Hello, everybody, and thank you very much, organizers, for inviting me to talk today. It's quite a short talk, hard to explain all intricacy of photosynthesis, photosynthetic machinery, but my specialty is harvesting light by all photosynthetic organisms on this planet. And I'm going to talk about efficiency and safety of this process. So our planet experiences a huge amount of energy from the sun, and from the point of view of our biosphere, this is the only source of energy that drives all life processes on our planet. So as we can see here, fields are covered with crops and they are green, and we know the green pigment is chlorophyll. The processes which really are involved in capturing light energy, or harvesting light, we call it, by photosynthetic pigments are fascinating, and there's an entire chain of events which leads to capture and storage of light energy. So it has been many decades in research on these two questions: how energy gets into the photosynthetic machinery through chlorophyll, and how it is being transduced into this formula, the major formula of photosynthesis, the reduction of CO2 at the expense of energy of light into the glucose, and evolution of oxygen. Oxygen we all breathe, all important formula. But let's disentangle this formula, and we can see it's not as simple as chemists would wish. The photosynthesis process actually evolves oxygen independently from fixation of CO2 from the chemical terms. And as a result of basically taking electrons from water, oxygen is evolved, and in the chain of light reactions of photosynthesis, intermediate carriers of energy, and ADPH and ATP are synthesized. Those are used in the dark for the synthesis of glucose. So we have a light and dark phases of photosynthesis. And here, where the oxygen evolution happens, a major player and only enzyme of nature, photosystem II, is involved, is responsible for evolution of oxygen and taking electrons from water, electrons charged with energy, and very important for redox reactions of photosynthesis. So let's dive into the nano world of photosynthetic machinery. Let's zoom in and we can see green grains, which are chloroplasts. As Darwin used to say, the grains of chlorophyll, they contain chlorophyll, and if we look closely at isolated grains, chloroplasts, we can see some structures even within. And then we look at an electron microscopy and we can see membranes packed inside of chloroplasts, quite a lot of them. The white ones are starch, accumulated starch granules. But what about these membranes? And these are the membranes where the light phase of photosynthesis actually takes place. They are flattened bags, basically called thylakoids. And in here, in these stacked so-called thylakoids, all photosynthetic machinery of photosystem 2 is localized. These stacks actually evolve oxygen. And if we have a look from the top of sliced membrane using so-called freeze fracture electron microscopy, we can see the particles of photosystem 2, these oxygen evolving particles of reaction centers of photosystem 2, which actually create oxygen for us and electrons for other reactions of photosynthesis. However, below we have so-called LHC2, a light harvesting complex, the major light harvesting complex, plenty of densely packed proteins there in comparison to the packing of photosystem 2, and those proteins contain most of the chlorophyll on our planet, and they are proteins which give energy to the photosystem 2. So if we look at schematic representation of stacked granules, stacked thylakoids, we can see that there are several complexes of photosynthesis. The photosystem 2 is here. It has a huge antenna, as I showed, a light harvesting complex, which collects energy of sun and delivers it to the reaction centers. And water splitting works, and protons are released into the thylakoid, so-called lumen. Then they are used to synthesize ATP, and electrons taken from water are used to reduce NADP to NADPH. Both of these substances, as you have seen before, they are used in the dark to synthesize glucose and reduce CO2. So what's about this light harvesting complexes? What's about this major vessel to carry chlorophylls? Actually, all chlorophylls of it absorb and give this energy to each other until it reaches reaction center. It's like domino effect, if you like. There is no substance transfer, just energy. As a result, we have excited electron of reaction center, and it can leave reaction center into redox stages of photosynthetic reactions leading to the synthesis of NADPH, and electrons from water are being replenished. Therefore, where the oxygen comes from, it's a simply byproduct of photosynthesis. Photosynthesis, strictly speaking, does not need it, but we need it very badly. So sometimes the photosynthetic light harvesting LHC2 complex is visualized like a funnel, the funnel of energy, the funnel which captures even very in shady environment. It captures sun energy, and it increases power of sun by 200 times. Light energy extremely dilute, particularly in shady environments. This light harvesting complex made it concentrate. So it's a concentrator of energy which focuses it after absorption onto reaction centers. So, but sometimes even antenna doesn't help, and we can see on the left that small seedlings, they reach for sun because their antennas are not sufficient to give them enough energy for growth in shaded environment of this greenhouse. By the way, it was in the house of Darwin, in his greenhouse, on a very shady day. And we see them all inclined towards the sun, and Darwin called this heliotropism, liking movement towards the sun. On the other hand, Darwin was one of the first who observed the paraheliotropism in high light photosynthetic leaves, photosynthetic organisms, they hide from light. Why is this? And if the leaf movement are restricted, like in these rosette types of plants, some plants survive whilst the others are not, and we have photosystem 2 efficiency dropped up to almost 0, and that spells death. That spells death. However, photosynthetic antenna are not only efficient harvester of light, but it's extremely clever and sophisticated system of proteins which carry chlorophylls, which can actually shut their efficiency and become less efficient. The excess of harvested energy can be dumped into heat in a very high light, preventing damage to the reaction centers and death of plant. And we can measure it using a gauge of this excitation pressure or excess absorbed energy by antenna in form of NPQ, or non-photochemical quench. So the protons produced as a result of electron transport and water splitting actually feed back, so it's called feedback mechanism, feed back into the antenna, and antenna becomes less efficient without removal of chlorophylls, without degradation, and it happens in time scale of seconds and minutes. So it is fast, extremely fast response for such a sedentary organism as high plant, for example. So what happened as a result with this antenna? As a result of delta pH of protons hitting the antenna in light become more clustered and aggregated. So there is a diffusion of all these proteins out of reaction centers to cluster together, and in this state it becomes inefficient. Well, the situation is not as simple as looking from just single particles of this major LHC2 complex or antenna. There are other proteins, minor antenna, PSBS, all a couple of pigments of carotenoids called xanthophyll cycle carotenoids, violoxanthine and zeaxanthin, which are all involved in this process of rearrangement of antenna under high light, segregation, clustering, and as a result in this state, excess energy dumped into heat. So on the left, you have efficient antenna in low light. In high light, it becomes safe. It becomes less efficient, but the amount of light that is absorbed in high light cannot be stomached by photosynthesis, and it's damaging. Therefore, it's dumped into heat. And that's what makes it safe. The outcomes of all this is that efficient and safe light harvesting depends upon protein composition in the membrane of photosynthesis. Membrane fluidity, so it's how easy these rearrangements and how quickly they can be established. Membrane permeability to protons, leakiness protons and other ions, protein lipid ratios and types of lipids which are present in the membrane. And this is all subject to sensitivity to temperature. And in current global climate change, because of rising CO2 levels, temperature can affect these whole processes and mainly has a detrimental effects. So what can we do about it? Well, first of all, we can act by establishing methods which analyze the trends on the whole plants. We can have this Fels color images of fluorescence, which we can monitor photosynthesis efficiency in plants. In parallel, we can monitor photoprotective efficiency in plants. For example, in the wild plants of Arabidopsis, we can see where photoprotection efficiency is high. It saves, it protects reaction centers of photosystem 2, and photosynthesis runs efficiently and strongly, whilst in plants, mutant plants, which are lacking one of the key carotenoids, key pigments of light harvesting antenna, the photoprotective efficiency is low, and we see the high impact of damage. So almost 50% of reaction centers disappears or dies. So, and this is early signs of damage. Some people can do engineering. They can manipulate this key protein PSBS and some pigments, xanthophyll cycle pigments, which previously I have shown to be involved in establishing safe dissipative state, so that these plants can track much faster fluctuating light. Fluctuating light can be even more damaging than light which is constantly of a high intensity. So shade and highlight, shade and highlight, so the antenna must track it much better and faster. And so what these people did, Kromdijk et al., they actually made the tobacco plants to monitor light fluctuations much more efficiently than the wild type tobacco plants. And that led to enhancement in plant growth and biomass, which is essential. And the final, we can raise awareness and teach the young generations that this is a time for photosynthesis research to act. And recent book, which we co-edited with Kristen Foyer, the chairman, and Professor Mercer from Nottingham, is a collection of articles which are focused on action of photosynthetic research and photosynthesis researchers in terms of changes of global climate and making plants more resilient to these changes, photosynthesis more robust and even more efficient in removing CO2 from our atmosphere. Thank you very much, and I'd like to acknowledge all my laboratory, which you can find on this link, and all lab members, current and previous ones which are there, and details of the research in my laboratory. And thank you very much.
Can you hear me, Alexander?
Yes.
My webcam won't work at the moment, but I would like to thank you very much for providing an introduction to photosynthesis, which is certainly one of the most, if not the most important biochemical processes on Earth, using CO2 to convert it into energy-rich molecules from which we all benefit, in addition to producing the oxygen we breathe. Right, I would like to move on now to our final invited speaker today. This is Professor Wolfram Weckert at the University of Vienna, Austria. Wolfram integrates system theoretical ideas with genome-scale molecular analysis using genomics, transcriptomics, proteomics, and metabolomics to understand and predict the genotype environment-phenotype relationship. Wolfram investigates plant, microbial, human, and animal systems. Working in the field of metabolomics since 2000, Wolfram has established metabolomics, proteomics, and phosphoproteomics platforms at the Max Planck Institute of Molecular Plant Physiology in Potsdam, Germany, for the Jeffries program of the Federal Ministry of Education and Research, Germany, and recently at the University of Vienna, Austria. In 2008, he moved as a full professor to the University of Vienna and founded the Department of Molecular Systems Biology, called MOSIS. Since 2013, he's the head of the newly founded Department of Ecogenomics and Systems Biology, combining ecosystemic approaches with high-throughput technologies such as genomics, proteomics, and metabolomics. Since 2015, he is founding chair of the Vienna Metabolomics Center. The Vienna Metabolomics Center integrates applied tools from environmental, biotechnological, and biomedical research questions. In the framework of the Vienna Metabolomics Center, the focus is on algorithms for data processing and integration, multivariate statistics, machine and deep learning, biochemical interpretation, structural elucidation of unknown metabolites, as well as metabolomics life science databases. The Weckert Lab develops genome-wide metabolomics and proteomics phosphoproteomics technologies as elementary systems biology techniques. Further research comprises data integration strategies by combining experimental approaches with multivariate statistics, pattern recognition, and modeling of metabolism. The Weckert Lab has also solved the biochemical Jacobian from metabolomics data using an inverse modeling approach. This approach combines statistical features of metabolomics data with metabolomics reconstruction and prediction from genome sequences and variable genotypes, and therefore establishes the systems analysis of the genotype environment-phenotype relationship. Wolfram uses these concepts in ecology, evolution and development, as well as biotechnology within the framework of green systems biology. And with this, I would like to hand over to Wolfram, please.
Thank you, Ilse, very much for this very kind introduction. Yeah, welcome everybody. So it's a great pleasure to share some results from my lab here. Just a short side remark. It's very hard to focus these days on science and having a war in a neighbor country. But we have to focus, of course. And I always remind myself to a famous German writer, Thomas Mann. He also got the Nobel Prize for Literature, and he had to escape from Germany in the Second World War, and he was very active in anti-war contributions by radio and writing. But at the same time, he decided, well, I'll focus on a completely different topic, and he sat down and started to write a very famous book on Joseph und seine Brüder, so Joseph and his brothers. It's a story of the Bible, and his book is now a kind of milestone in German literature. The war is over, the book is still there and is important for us. So I hope somehow we can really solve this problem very soon and have a better world afterwards. Okay, now I focus on research, okay? So it's about crops in a changing climate, of course. I will focus now on crop plants, and I will introduce some ideas on how systems biology is applied in germplasm collections, and with a focus on the understanding of intraspecific crop plant adaptation in the climate crisis for climate change mitigation. So this is also an option, of course. We heard very nice results already on a net primary production processes, on photosynthesis, but you can think also about crop plants and agriculture as a climate change mitigation procedure. So now it's hopefully going on. Right, we start with a hypothesis now that agriculture contributes to the mitigation of climate change due to its nature as carbon sequestration process. Of course, and this was nicely introduced by Alexander, photosynthesis and CO2 fixation. However, the current status is that we have a net, we have a net greenhouse gas emission instead of sequestration. So this is due to many observations. First of all, the harsh climate reduces plant growth and productivity, and this leads to inefficient CO2 neutral biomass and food production. I will come to this later. Secondly, there is a high demand on industrial nitrogen fertilizer for productivity. It's a huge amount of nitrogen and anthropogenic. It really influences the global nitrogen cycle on Earth by the anthropogenic influence, by the Haber-Bosch method. But on the other hand, there's a low nitrogen use efficiency due to losses in the soil. So this is called the nitrification process. And at the same time, you have, due to this process, also greenhouse gas emissions by the denitrification process. I will explain this in more detail, and we can actually address these questions. Another big part is, of course, food production and consumption. These are industrial processes, and altogether, you have really a net greenhouse gas emission. But you can, of course, work on this. So this is not a new idea, of course. This is quite an old idea. It's in the framework of agroecology, and I think this is a key to agricultural resilience and ecosystem recovery. So there's a very nice seminal work by Conway in the 80s, and he summarized the major topics of agroecosystems. It's productivity, stability, sustainability, and equability. And these 4 points are fundamental properties of any complex system. Actually, they are derived from analysis of systems. So, and this is a scientific theory, it's systems theory. And so this agroecology is really bound to systems theory and also to systems biology. And it's very important that you start to think about systems theory in this context also. So the FAO has here a very good definition of agroecology. So you have efficiency, resilience of agriculture, but you have also human social values and culture and food traditions, and all this goes into synergies and feedback into the diversity on Earth. And this kind of diversity, biodiversity, is genetic variation. It's natural genetic variation. So it's basically what we nowadays call germplasm genomics. So in the field of applied science, with crop plants, we really assemble whole germplasm collections with genome data. What does it mean actually? So you all are aware of this next generation sequencing. You can really sequence organisms in days nowadays. And so we have really an exponential growth of genomes available. And this, of course, holds true for crop plants as well, right? So here, rice or a potato genome or a tomato genome. And it is the case that we do not only sequence one individual of these cultivars, but we sequence thousands of these individuals. So we have really a large collection of genomes available. So here, this is an example with Arabidopsis thaliana. We are part of this consortium, and we recently sequenced 1,135 genomes from one species. This is now Arabidopsis thaliana. It's just one species. Therefore, it is intra-specific variation, which we see here. And we have ecotypes from North Sweden to South Spain. And you can imagine that these ecotypes have completely different phenotypes and also adaptation processes to climate. And now we can correlate the genomic variations, so-called polymorphisms, to these climate adaptation processes, and we can identify some basic fundamental processes which are important for the adaptation. We apply this whole approach also in crop plants. So here is an example where we work together with a non-profit institute with a large germplasm collection on pearl millet. It's a millet, it's a staple food crop in low-income for low-income farmers in the Asian region. And here you see we sequenced many cultivated genotypes, but also wild types from the African region. And now we have all this information available. So now we can learn a lot from this genome data, but the major question is, can we actually predict growth, physiology, stress phenotype, and yield, quality, and also adaptation to climate change? This is a major question, right? And so we recently assembled a large collection of so-called genome-wide association studies, GWAS, where they correlated genome variations, so polymorphisms in the genome, to the phenotypic variants. And they were able to explain phenotypic variants by the genome only genome information, but it was in the range of 10 to 40%. So actually 60 to 80, 90% of phenotypic variants is not explained by genome data. So how can we understand this complexity now? Or why is it so? So the genome encodes the proteome, and here you have already an amplification of protein species. So from a typical genome, you have millions of proteins encoded. By post-translation modifications, you have a further amplification. By metabolome, the metabolome is a further amplification of molecular complexity. You have all these interactions, and this all generates the omics cascade. And this is basically a pan-omics relation, where the genotype is explained by all these interactions of this complex molecular network. So the genotype-phenotype relation is explained by this interaction. And this leads to this point that we cannot explain the phenotype alone with the genome data. So for this reason, we developed a pan-omics platform where you integrate further levels of molecular analysis like proteomics, metabolomics, and also integrate genome scale modeling. And we applied this to various different crop plants, tomato, potato, cereals like pearl millet, wheat, barley, but also legumes, for instance, like chickpea. And now what we see indeed emerging more and more, that we can explain more and more phenotypic variants by integrating the genome data with the further molecular data like proteomics, metabolomics, transcriptomics. And this would allow then in the future to integrate this data and to select for specific cultivars or wild types from these germplasm collections for molecular assisted breeding or even genome editing to produce elite lines which are adapted to various different environmental stressors. So I come now to the examples. How do we apply these platforms? So there's a first point that we see plant growth reduction by strong environmental pressures, for instance, drought stress. So how can we address this? So we now go into the germplasm collection, and these germplasm collections offer really an opportunity to identify climate smart crops with high stress resilience. We published this recently, and we identified two different genotypes. For instance, one is sensitive genotype and a tolerant genotype in this pearl millet germplasm collection. And by applying our omics platform, our pan-omics platform, we identified lots of molecular markers which are really in opposite direction in these two genotypes. So in the tolerant genotype under drought stress, we see a maintenance of the photosystem, photosynthetic machinery. And we see actually in comparison with the sensitive genotype, we see that they can cope much better with oxidative stress processes. So, and at the same time, we have a higher yield also in the tolerant genotype. And this is a so-called stay green phenotype. So the leaves stay green under drought stress. And now we have all these markers which you can feed into this workflow and provide then the breeding processes with special molecular markers where we can look for these specific stay green phenotypes, which are much better adapted to drought stress and other climate changes. So in the second example, I will address this problem of nitrogen fertilization. And here we have also opportunities using germplasm collection to improve this process. So first of all, you have to understand what's going on in this process of nitrogen fertilization. So you have a plant and the root system, and you can understand plant physiology only in the context of the root system. And now you have the nitrification process in the soil by bacteria and archaea, and they take actually the nitrogen fertilizer ammonium and oxidize this fertilizer to nitrate. Nitrate by these two processes, by ammonium oxygenase and hydroxylamine oxidoreductase. And these processes are catalyzed by bacteria. This leads to high amounts of nitrate, which is then leaching from the soil. So nitrate leaches easily from the soil by rain. So you lose actually, the more fertilizer you put on the soil, you lose more and more nitrate here. At the same time, the denitrification process is stimulated by nitrate, and this leads then to nitrous oxide, and this actually is a very strong greenhouse gas. So this is released from the soil. So if you put now nitrogen fertilizer on the soil, you have these processes, and you actually have a very low nitrogen use efficiency by the plant due to these processes. But there are so-called biological nitrification inhibitors released by the root system of the plant. The plant can actually control this process by these inhibitors and can inhibit these processes. And now we can go into the germplasm collection and look for many different cultivars or even wild types which have a very strong BNI activity. So we did this recently also with pearl millet plants. We investigated them also under drought stress, and then we investigated the drought stress, so the root exudation by these genotypes. And indeed, we identified different genotypes, different root exudate diversity. So these genotypes really are very different in the root exudation process. And again, remind yourself, this is intra-specific variation. So this is already a very exciting result. And we found indeed a genotype which has quite a pronounced biological nidification inhibition here under stress conditions. So indeed, we can now screen the whole germplasm collection for these processes. What does it mean now in consequence? So these root exudates from the plant release biological nidification inhibitors. So this actually decreases the nitrification process and increases then the nitrogen use efficiency for the plant. And at the same time, you decrease the release of greenhouse gases. And this is how we want to approach, by using germplasm collections, this overall process of agriculture and to improve the greenhouse gas emission efficiency to mitigate climate change. Okay, so the PanOmics platform is able to explain up to 80 or 90% of phenotypic variation. So it's really important to implement these data into so-called genome prediction tools nowadays. This information is necessary to explore the full power of molecular marker assisted breeding and genome editing. And this platform allows to focus on climate smart crops and agroecology as a process to mitigate climate change. Thank you very much. We have, of course, these are collaborations with non-profit organizations, so ICRISAT, and then it's indeed for the benefit of the world, I hope very much. And I would like to thank also my collaborators, Arindam Ghatak and Palak Chaturvedi, very much here for their contribution. Thank you very much for your attention.
Thank you very much, Wolfram. For starting with very kind words on the extremely worrying situation in the Ukraine at a moment when we're all lost for words, really. But of course, also for highlighting the importance of omics techniques and systems biology in plant science, and more generally also for highlighting the pivotal role of plants for our planet. Right, at this moment in time, I would like to thank all three speakers again. And I would like to mention that we can now welcome questions for our speakers. And if you have a question, please type in the question box as shown in the image on the screen. At this point, I would like to thank the speakers again and hand over to Christine, please.
Oh, thank you very much, Ilse. Thank you also from me to all the speakers. I really enjoyed all the talks and gave us a very diverse outlook on the topic. So I will go straight into the questions that I can see. And I believe the first question is for Professor Ruban. And it says, is there any alternative process performed by plants to trap energy?
Sorry?
I think so. I think other than photosynthesis, I think this is what it means. Are plants able to trap energy in other ways?
I don't know any other way to trap energy, but through photosynthesis by plants.
It's true. I agree, Sasha, but you can grow plants on glucose. I don't know how long they survive, though, do you?
No, that's, I don't know. They will not grow. But yeah, that's the only way is photosynthesis.
Yeah, exactly. I agree with you. So moving on, I have a question for Dr. Pan. In the UK, politicians are encouraging everyone to plant trees to combat CO2 emissions. From your data, I believe that grasslands are better than deciduous broadleaf forests. Can you please comment on that?
Yeah, I guess now, you know, we do see now that not those dense forests and tropical forests, their ability to sequestrate carbon seems like a little bit kind of the decrease because, you know, I said the saturation of the forest canopy. So, but we see the temperate zone seems has kind of doing pretty well, you know, given the reason I mentioned. So if you plant trees, of course, they can help to kind of sequestrate the carbon. But I think more important things based on actually we are now doing some of that global forest carbon, estimate again, to update our study 10 years ago. So you do see that how important to stop deforestation in the tropical region, because the tropical forest, this is much the powerful, useful than you plant trees in some less suitable area place to plant trees. I think because I hear the Professor Ruban and the Professor Wuhan, they talk about agriculture. These are our study. You also see agriculture land have kind of the increase a lot in their productivity because of those high CO2. But we do think those ecosystems, they cannot hold the carbon very long time. Those carbon will be, from those products will be released. Not like the forest, they can, in some way, they can keep those carbon. So I don't know if I really answered the question, but I think to plant trees definitely helpful. But I think stop the deforestation in the tropical region is more important and urgent things we need to concern.
Thank you very much, Dr. Pan. I can't see another question at the moment. Does anybody else want to write a question in the question box? Okay, maybe we don't have any more questions at this moment in time. So maybe we can finish here. And with Ilse again, we'll thank everybody and remind you again, please contribute to the plant collection. The details are on the website. And please, if you want to ask more questions for our speakers, I'm sure they'd be happy to answer questions by email. So without further ado, I think I will say thank you and goodbye. Ilse, do you want to have the last word?
No, I would just like to thank everybody for attending and and thank the speakers again for giving a great overview and introduction into the topic. Yeah, and to everybody, please just consider submitting your work to the special issue of the Biochemistry Journal. And yeah, thank you and goodbye.
Yeah, thank you. Goodbye.